All right, ladies and gentlemen, today we're going to talk about chapter 26 and really sort of take into consideration all of this evolutionary history of organisms and how do we start diagramming that, explaining it, putting data together to actually tie individual organisms to their common ancestry. So, what we're really talking about here is this idea of phylogeny and systematics. So, let's just start with what those two terms mean. When we talk about phylogeny, we're talking about the evolutionary history of a species or a group of related species. So, where are those branching points? What are the things that they share in common? What are the things that they have diverged from each other as a result of different environmental conditions? And then systematics is just the discipline that actually classifies organisms and determines those evolutionary relationships. So systematics is really utilizing all sorts of resources and data to start tying together the phylogeny of different groups of organisms. So things from fossil record data to molecular and genetic data. So how are the molecules and the DNA, RNA, proteins are usually the three big ones. What sort of data can we infer from things that they share to actually backtrack and trace where those divergence points might have occurred during evolutionary history? So, taxonomy is probably the most commonly known way that we classify organisms. It's that ordered division of categories based on a set of characteristics. And this goes all the way back to Carolus Linnaeus and the first initial classification scheme. It's this hierarchical classification that ultimately ends in what we know as binomial nomenclature, and that's the species scientific name, right? And that accounts for both their genus and then their specific species. But that backtracks all the way up to the family, the orders, the classes, the phyla, the kingdoms. And now ultimately we also use that domain system as well. So again, it ultimately gets down to their scientific name, right? Binomial literally translates into two names. So it's a two-name naming system that we utilize to classify organisms. That first part being their genus. The second part, most of you have probably learned it as genus then species, but you may also come across this term called their specific epithet. Use it interchangeably with species, and most people will know what you're talking about. Just some basic housekeeping when we write scientific names for organisms, right? That first letter for their genus should always be capitalized. The species is always lowercase. And if you're typing it out, then the entire species name, the entire genus and species should be italicized. If you happen to be handwriting it, then we would underline both that genus and species. So again, these taxonomic groups go from very broad all the way up to domains where we're just talking about their different internal organization of their cells. Are they eukaryotic or prokaryotic? All the way up to that specific species that you're dealing with. In this case, it happens to be this leopard. So taxons are those taxonomic units at each level. So we can talk about the taxon for mammals, and the class happened to be Mammalia, right? The broader the taxa, the less useful it is from an evolutionary standpoint in order to be comparing their lineages. So that doesn't necessarily mean anything to their evolutionary history. It can, but you've got to dig in a little deeper. So basically what I'm getting at is something like this example here, where an order of snails has less genetic diversity than the order of mammals. So if I'm comparing between the same orders, or I should say the same taxon, so the same level, so in this case the order, one order of a set of organisms doesn't necessarily relate to another. So point is you can compare up and down in terms of relationships to each other, but not necessarily within the same taxon. So hopefully that makes sense.